Good morning, everybody. Welcome back to daily lectures at New Age Cosmetic White EdTech Healthcare Consultancy. I'm Dr. Bobby. Today we're on the subject of molecular biotechnology. We're specifically on the subject of nucleic acid therapies. We're on the subtopic of aptamers. Now, when we talk about aptamers, these are nucleic acid sequences, RNA or DNA, that bind tightly to proteins, amino acids, drugs, or other molecules. They are typically 15 to 40 nucleotides long and have highly organized secondary and tertiary structures and bind with high affinity, 10 raised to minus 12, less than the dissociation constant, less than 10 raised to minus 9, where Kd is the dissociation constant to the target molecules. Aptamers are attractive as potential therapeutic agents because of their high specificity, relative ease of production, low or no immunogenicity, and long-term stability. Aptamers that are directed against specific targets are typically selected by a procedure known as SELEX, systematic evolution of ligands, by exponential enrichment, in which DNA or RNA ligands that bind to the target molecule are selectively enriched. In this procedure, a random DNA sequence is cloned between two particular DNA sequences. The 3' region contains an attachment site for reverse transcriptase primers, and the 5' region contains an attachment site for a polymerase chain reaction PCR primer. The double-stranded DNA is converted to RNA using T7 RNA polymerase. The SELEX procedure combines several rounds of binding, partitioning, and amplification of selected nucleotide sequences from an initial pull-up of about 10 raised to 16 nucleotide sequence variants. The end result of this procedure is the selection of aptamers that bind to target molecules with high affinity. Ultimately, the SELEX procedure yields one or just a few unique nucleic acid sequences from an original mixture with high affinity for the target molecule. To make aptamers less sensitive to nuclease digestion, OH residues at the 2' position of purines may be replaced with 2' O-methyl residues. In addition, aptamers may be kept at their 3' end with a deoxythymidine residue. Table 11.1 lists some of the proteins against which aptamers have been generated, as well as a range of affinities of the aptamer for the target protein. An aptamer known as Pegaptanib received approval from the US FDA in December 2004 for use as a human therapeutic agent. Pegaptanib is a 13-nucleotide long aptamer that targets vascular endothelial growth factor VEGF and binds to the protein with extremely high affinity at dissociation constant equal to 0.05 nm. This secreted protein promotes the growth of new blood vessels by stimulating endothelial cells that not only form the walls of the blood vessels but also transport nutrients and oxygen to the tissues. When retinal and pigment epithelial cells begin to seize from lack of nutrition ischemia, VEGF acts to stimulate the synthesis of new blood vessels called neovascularization. However, this process is imperfect and often the blood vessels do not form properly, so that leakage results, causing scarring in the macular region of the retina with the eventual loss of central vision. These physiological changes contribute to age-related macular degeneration, a leading cause of blindness. Pegaptanib was selected to bind to one of the 4 isoforms of VEGF. Pegaptanib. Excuse me. To form VEGF 165. Right, that is responsible for age-related macular degeneration. The drug is injected directly into the eye every 6 weeks or about 9 times a year. All forms of VEGF have a receptor binding domain, but only VEGF 165 has a heparin binding domain. So Pegaptanib is a new type of therapeutic agent with an unusual specificity that can effectively suppress age-related macular degeneration. To date, approximately 95% of the patients receiving Pegaptanib were 65 years of age or older. The safety of aptamers used in clinical trials is a concern, especially when the optimal dose of a particular aptamer is not known. One way to overcome this problem is through the use of aptamer antidotes. These molecules consist of short oligonucleotides whose sequences are complementary to the aptamers being tested. When antidotes are added to aptamers, they hybridize to the aptamers and inhibit their binding to the clinical target. Interfering RNase principles. The addition of double-stranded RNA to animal and plant cells reduces the expression of the gene from which the double-stranded RNA sequence is derived. This gene silencing, which specifically reduces the concentration of a target mRNA by up to 90%, is reversible. Since there is no change in the target cell's DNA, this phenomenon has been termed RNA interference, or RNAi, and occurs naturally in virtually all eukaryotic organisms. RNAi appears to be the same phenomenon as gene silencing in animals or co-suppression in plants. And although all of its biological roles remain to be established, RNAi protect both animals and plants from viruses and from the accumulation of transposons. A working model for RNAi has been formulated based on experimental analysis. Following the introduction of a double-stranded RNA molecule into a cell, the double-stranded RNA is cleaved by the RNase 3 like enzyme dicer into single-stranded pieces of RNA, approximately 21 to 23 nucleotides in length, that have been called small interfering RNAs or siRNAs. The antisense strand of a siRNA incorporated into an RNA inducing silencing complex RISC that binds and then cleaves the mRNA. The specific binding of the siRNA, that silencing RNA, to the mRNA that occurs is based on the complementarity of the 2 RNA sequences. The site of cleavage of the targeted mRNA is between nucleotides 10 and 11 relative to the 5' end of the siRNA antisense guide strand. Consistent with this model, the transfection of mammalian cells in culture with duplexes of 21 nucleotide RNA can also mediate an RNAi. Despite the fact that many aspects of RNAi are still not completely understood, it could form the basis for new therapeutic agents. The use of short RNA duplexes may eventually provide an alternative approach to the use of antisense oligonucleotides or ribosomes. Now, the phenomenon of RNAi is expected to facilitate the development of a wide range of antiviral compounds and therapies utilizing specific silencing RNAs delivered to the appropriate target cell. Similarly, the expression of endogenous eukaryotic genes may be inhibited by a plasmid-driven expression of short-happen RNAs, that is, shRNAs are short-happen RNAs, which are similar in structure to the microRNAs that often normally regulate gene expression in eukaryotic cells. In fact, there are a number of reports of the use of either siRNA or shRNA to suppress virus replication. In tissue culture, moreover, it has been successfully demonstrated that RNAi is effective and inviolable with mice, suggesting that, in principle, all viruses may be inactivated by RNAi, independently of how an siRNA or shRNA, that is, silencing RNA or short-happen RNA, is introduced into a cell. It may have non-specific effects. For example, introduction of these molecules may inadvertently activate innate cellular immune responses, such as the interferon response. In addition, siRNA or shRNA may also be complementary to non-target mRNAs. However, several experimental approaches may be utilized to avoid these problems. Off-target effects are most often observed when the siRNA or shRNA concentration is less than or equal to 100 nm. By lowering the concentration as much as possible, often up to 20 nm or less, off-target effects are often avoided. 2. Since the interferon response can be induced by double-stranded RNAs with as few as 11 BP that are perfectly complementary, siRNA or shRNA is designed to contain at least one nucleotide bulge where the bases on opposing strands are non-complementary near the center of the molecule, typically around 21 BP. 3. Since siRNA or shRNA can exert a toxic effect when it contains a sequence 5' UGGC' 3', the sequence should be avoided. 4. Blunt-ended 27 BP RNA duplexes or 29 BP shRNAs with two nucleotides overhanging at the 3' end are much more potent inducers of RNAi than 21 more siRNAs. The greater level of effectiveness of the slightly longer RNAs may reflect the fact that they are first bound and cleaved by DICER, which facilitates the entry into the RISC. Using these slightly long RNAs at lower concentrations should avoid side reactions associated with 21 more siRNAs. Unlike siRNAs, shRNAs are expressed in vivo as part of a genetic construct that includes a promoter sequence. This means that shRNAs need to be introduced by using strategies different than those used with siRNAs. Thus, shRNAs are typically delivered to their target cells by using viral vectors. Viral vectors that integrate into the chromosomal DNA are generally used when persistent long-term knockdown of gene expression is desired. The most popular choice is lentiviruses. However, with the use of all virus-based vectors, there are serious safety concerns that need to be addressed, and the type of promoter that is used also needs to be optimized. Applications. Interfering RNAs have already been found to have widespread use as tools in research that is directed towards understanding how gene expression is regulated in natural systems. One company that specializes in producing RNAi directed against human mRNAs advertises for each target human gene. We provide 4 plasmids, each with a different short-happen RNAi sequence. shRNA the experimentally verified design algorithm minimizes the risk of off-target effects and ensures maximum knockdown. There's a claim at least one of the 4 shRNA plasmids will reduce the target mRNA levels in the target transfected cells by about by greater than 70%. With the ready availability of human shRNA libraries, new insights and understanding of many fundamental and disease processes should be rapidly forthcoming. It is hoped that this in turn will lead to a variety of new therapeutic agents and approaches. In addition to the many reports of successful modifications of the gene, expression of cells in culture with RNAi, there are an increasing number of reports of the in vivo effectiveness of RNAi. By 2008, siRNAs against a wide range of proteins, viruses, and diseases had been successfully expressed in mice, including siRNAs directed against herpes simplex virus type 2, herpes B virus, hepatitis B virus, hepatitis C virus, Huntington disease, metastatic Ewing's sarcoma, a form of cancer, respiratory syncytia virus, hepatic cancer, transforming growth factor receptor 2, severe acute respiratory syndrome, hemoxygenase 1, keratinocyte-derived chemokine, tumor necrosis, factor alpha, and human epidermal growth factor receptor. Now, in addition, in 2008, 3 different RNAi therapeutics were being tested in clinical trials. All 3 of these therapeutics target VEGF or its receptor, a cause of age-related macular degeneration. And at that time, all had successfully completed either phase 1 or phase 2 clinical trials. And now, into the antibody genes. A major problem with aquaculture is the loss of fish due to bacterial and viral infections. The chemical treatments that protect fish against these disease-causing pathogens are extremely costly and not particularly effective. One way to address this problem is to develop fish that synthesize protective antibodies against particular pathogens. The impetus for this approach came from the observation that rainbow trout could be protected against hemorrhagic septicemia virus by passive immunization through the injection of a monoclonal antibody against the G protein from the virus. Subsequently, a gene encoding a single-chain antibody directed against the hemorrhagic septicemia virus G protein was synthesized. The synthetic gene-encoded variable regions from the mouse LNet chains of an anti-G protein monoclonal antibody with a human antibody constant domain fused to the 3' end of the construct. To mediate secretion in fish cells, the DNA was fused to the gene sequencing encoding the signal sequence. And of a rainbow trout transforming growth factor beta. The DNA construct was inserted into a eukaryotic expression vector under the transcriptional control of a constitutive cytomegalovirus promoter, cytomegalovirus promoter. The clone gene vector construct was injected into the circulatory system of rainbow trout when the fish were challenged with the hemorrhagic septicemia virus. 11 days after injection of the DNA construct, nearly all of them survived, whereas the control fish did not. 39 days after the DNA injection, the plasma concentration of the single-chain antibody remained at a high level. Thus, instead of administering a purified antibody to provide an animal with passive immunity, it is possible to inject an animal with DNA encoding the antibody. Giving an animal the ability to synthesize a specific antibody may be more efficient than depending upon the animal's immune system to produce a similar antibody. However, since it may not be practical to inject large numbers of fish, the next step will be to create transgenic fish with antibody genes that confer passive immunity to various diseases. Nucleic acid delivery. The ultimate effectiveness of any therapeutic agent depends upon the ability to deliver the agent to the tissues where it is required. Systemic introduction of a therapeutic agent often leads to the accumulation of very high levels in tissues where the agent is not required, and sometimes results in serious side effects. To this end, viral vectors that deliver small nucleic acids to specific cellular targets have been developed. However, although virus-based gene delivery has been successful, a number of safety concerns have arisen in regard to the use of these vectors. Several approaches have recently been developed as an alternative to virus-based systems, as well as to the systemic introduction of target nucleic acids. There are several methods that have been used to deliver relatively small nucleic acids to animal cells. They include 1. Intravenous injection, 2. Local injection at the site of the pathology, 3. Packaging into cationic liposomes, physical methods like electroporation, sonoporation, or hydrodynamic pressure, and 5. A number of systems in which the nucleic acid is chemically conjugated to another molecule. Human gene therapy. Most of the experience of managing genetic disease have been gathered from inherited inborn errors of metabolism and to a lesser extent from disorders with defective structural proteins. The strategies for treating genetic disorders include restrictive or supplemental diets, inhibition of enzyme reactions to prevent accumulation of toxic molecules, removal of toxic molecules, replacement of defective or absent proteins, restoration of protein activity, selective protein removal, organ and bone marrow transplantation, nucleic acid-based therapies. With the development of recombinant DNA technology, large numbers of enzymes and structural proteins are now available for therapeutic use. Infusions of beta-glucosidase, beta-galactosidase, alpha-L-iduronidase, and adenosine deaminase ADA. In clinical trials have significantly reduced the adverse effects of Gaucher's disease, Fabry disease, mucopolysaccharidosis 1, and severe combined immunodeficiency disorder (SCID), respectively. This type of treatment, called enzyme replacement therapy, works well when either the enzyme or the structural protein replacement therapy is delivered to its biological site of action through the bloodstream. In the 1940s, when it was discovered that a gene from one strain of bacteria could be transferred to and expressed in another strain, researchers have contemplated the possibility that human genetic diseases might be cured in an analogous manner. Introduction of a novel gene into a cell with a defective gene ought to correct the disorder because the transferred I'll just be back in a minute. Gene provides the required gene product. In theory, gene therapy should provide persistent in vivo treatment either in the tissue that is primarily affected by a gene mutation or in deficient cells that acquire a recombinant protein from distant cells that release therapeutic protein into the circulatory system. And although gene therapy was intended as a cure for genetic disorders, gene products can also be used to treat cancers, infections, and various degenerative disorders. And now, before we go into the next portion, let's just finish up this table that talks of strategies for treating genetic disorders. So it was specially formulated diets, restricted diet to lower the level of toxic molecules, supplemental diet to replace a metabolic deficiency, inhibition of enzyme reactions, enzyme inhibitor to prevent the accumulation of a toxic molecule by blocking a step in a metabolic pathway that precedes the reaction with a defective enzyme. Removal of toxic molecules, dialysis, removal of excessive cations, chelation, facilitation of excretion by binding a toxic molecule to a lower molecular weight compound, replacement of defective or missing products, enzyme replacement therapy, protein replacement therapy, cofactor supplementation, alteration of defective protein by small molecules, restoration of partial protein function, directed proteolytic degradation for a defective protein, and transplantation is for a replacement of a non-functional organ transplantation, providing a required protein synthesized by blood cells, bone marrow transplantation, gene therapy, rectification of a genetic defect with a functional gene, nucleic acid therapy, blocking translation of mRNAs from a mutant gene with an oligonucleotide antisense ribosome, correction of a gene mutation with an oligonucleotide. This gene provides the required gene product. In theory, gene therapy should be provided a persistent in vivo treatment, which is already explained. Now, in the 1990s, after exhaustive reviews by many different regulating panels in the United States, the first human gene therapy trial was initiated. 2 young girls with ADS deficient SCID received large doses of their own cells that have been engineered to carry functional ADH gene. In both instances, the adverse symptoms were alleviated, indicating that this form of therapy is feasible. There have been drawbacks as well. And although in the broadest sense the concept of human somatic cell gene therapy is straightforward, there are a number of critical biological considerations. For example, how will the cells that are to be targeted for correction be accessed? How will the therapeutic remedial gene be delivered? What proportion of the target cells must acquire the input gene to counteract the disease? Does transcription of the input gene need to be precisely regulated to be effective? Will overexpression of the input gene cause alternative physiological problems? Will the cells with the input gene be maintained indefinitely, or will repeated treatments be required? Much of the research effort in human gene therapy has been directed to developing efficient and non-immunogenic systems that deliver therapeutic gene to a specific cell type. Both viral and non-viral strategies have been examined in detail. Different types of viruses, including retrovirus, adenovirus, adeno-associated virus, herpes simplex virus, and vaccinia virus, have been engineered as gene transfer vectors. A virus-mediated gene delivery system uses the cell receptor recognition system of the virus for binding to a specific cell type, which is followed by the internalization of the vector DNA and its transport to the cell nucleus. Some viruses have mechanisms for integration of the vector DNA transfers into a chromosomal site, and with others, and input DNA is maintained as an extra chromosomal element. Since high efficiency of vector DNA transfer is important, the administered sample of viruses should contain mostly vector viruses with very few and preferably no infectious virus particles. To meet this goal, packaging cell lines were constructed for some virus vectors. Those cultured cells carry genes that express viral proteins that are necessary for the formation of virus particles, but are not capable of producing replication-competent infectious viruses. Autotransfection of a packaging cell line with a vector construct that is equivalent to the length of the wild-type virus genome and that carries the appropriate DNA sequence for packaging into a virus particle. The input DNA is replicated, assembled into viruses, and released into the cell medium. The non-infectious vector viruses are concentrated and prepared for use. Packaging cell lines have been devised for retrovirus vectors and other viral delivery systems. In some cases, disarmed viral and vector DNAs are co-transfected and only viruses with vector DNA are produced. In other instances, protocols allow the formation of both vector and infectious viruses with the separation of the two types of viruses before use. There are advantages and disadvantages to each of the major viral gene delivery systems. Some vector viruses transduce DNA at high efficiencies, but the size of the insert is limited. Retrovirus vectors can infect only dividing cells, which without genetic modification makes them ineligible for treating disorders of non-dividing cells. Some viruses lack cell specificity. However, this particular shortcoming has been countered by designing viruses with cell-specific receptor binding sites. Many vector viruses are immunogenic, which nullifies repeated treatment with the same virus strain. To overcome this problem, vector viruses with different antigenic determinants may be used for successive treatments. Currently, third and fourth generation varieties of vector viruses are being developed, which I think should be far ahead now that this edition is the fourth edition, with distinctive features for specific illnesses. Some successful gene therapy experiments using viral vectors have been reported. And in phase 1 clinical trials, a number of patients have been treated with recombinant adeno-associated virus vector carrying a human cDNA encoding retinal pigment epithelium-specific 65 kDa protein, RPE65. The protein encoded by the RPE65 gene is an important part of the visual cycle, forming part of a pathway that regenerates the visual pigment after exposure to light. Individuals who lack this protein become deficient in Levin's retinal, and their rod photoreceptors are unable to respond to light, eventually leading to blindness. Prior to undertaking these experiments, a strain of bad dogs, the Bayard dogs, with a spontaneous defect in the RPE65 gene was successfully treated with the restoration of visual function in the treated dogs. In all instances, the recombinant viral vector carrying the restorative gene was introduced by surgery of the eye into the subretinal space. Although the phase 1 human experiments were aimed only at determining the safety of this treatment, an improvement in the vision of 4 of the 6 patients was observed. This encouraging result notwithstanding a longer follow-up. This is a production of packaged retrovirus vector RNA. An illustration of that. The packaging cell line has 2 separate retroviral gene regions on its chromosomes. One contains the GAG gene, the other contains the Pol and N genes. In each, these inserts transcription is driven by sequences within the 5' long terminal repeat, 5' LTR region. Both viruses' DNA segments lack the encapsulation sequence psi that is required for packaging a retroviral genome into a viral capsid. Now, the period and clinical trials with many more subjects are required before this procedure can gain FDA approval. However, on the positive side, if the procedure is used to treat younger patients with less severe disease, greater improvement in visual function might be expected. Notwithstanding the advantages of viral vectors, they are often immunogenic, costly to maintain, and difficult to produce on a large scale without high-level expertise. Consequently, various non-viral gene transfer systems have been devised. The least complicated non-viral gene delivery system is the introduction of pure naked DNA constructs directly into the cells of a target tissue. With plasmid DNA injected into mouse skeletal muscle, some of the cells took up the DNA and a reported gene was expressed for more than 50 days. However, this approach is limited to accessible tissues and requires large amounts of DNA. Pure DNA constructs that cover the surface of 1 to 3 micrometer diameter gold particles have been propelled with a gene gun into skin cells, as shown in chapter 18, into subcutaneous tumor cells. Therapeutic genes delivered in this way were expressed in the targeted tissues surrounding a DNA construct, with artificial lipid layers that form a lipid sphere with an aqueous core. Liposome facilitates the passage of the DNA through a cell membrane. To avoid degradation of introduced DNA molecular conjugates have been developed with this approach. Poly-L-lysine is chemically linked, conjugated to a molecule that binds to a specific cell receptor. Next, DNA is added and combines with the poly-L-lysine to form a tightly compacted, twisted, solid ring with the cell receptor binding sites arrayed on the outside of the DNA molecule. Conjugate, the complex is bound exclusively to the specified cells, but the frequency of transfection was low. To remedy this problem, in addition to a cell receptor binding amino acid sequence, other short amino acid sequences that facilitate cell membrane fusion and internalization of the DNA molecular conjugate, and both protect the DNA from degradation and direct it to the cell nucleus, have been combined into a single polypeptide. The addition of such a multifunction protein to a DNA molecular conjugate could enhance the efficiency of transfection. The current non-viral gene delivery systems have two major limitations. That is, one, the frequency of transfection is often too low to create a therapeutic effect, and two, the duration of therapeutic gene expression is too brief to provide an effective treatment. A human artificial chromosome HAC could be an exciting therapeutic vector. The DNA carrying capacity would be very large, which would allow the inclusion of several genes, each with a complete set of regulatory elements. This type of vector should have long-term stability and sustained expression of therapeutic genes with either a proliferating or a quiescent target cell. HACs, also called human-engineered chromosomes, have been created in two ways. First, HACs were assembled by ligation of individual chromosome components, including the chromosome ends, telomeres, centromere, and origins of replication. Telomere and centromere sequences were mixed with high molecular weight human DNA that had both origins of replication and a selectable gene marker in the presence of ligase. Cells were transfected with the DNA from the ligation mixture, and those with HACs were selected and maintained a second method of forming of HAC entails paring down an existing human chromosome by deleting material from within each chromosome arm to form a mini chromosome. HACs have been formed that range in size from 0.7 to 400 megabases. However, before HACs are used for gene therapy, certain issues must be addressed. For example, will HACs efficiently introduced into the nuclei of target cells? Will effective levels of therapeutic gene expression be maintained for extended periods of time? Before therapeutic genes are introduced in human beings, the efficacy of using a particular gene along with a specific delivery system is tested on small animals, typically mice. This is intended to ensure that not only that the added gene relieves a particular ailment, but also that there are no unexpected side effects that occur as a consequence of the treatment. Recently, researchers reported the administration of a gene to dystrophic and normal mice that helped them to increase both muscle biomass and strength. The growth factor myostatin plays a critical role in regulating skeletal muscle mass. It negatively regulates both the numbers of myofibers formed in development and the postnatal growth of muscles. It was previously suggested that a number of neuromuscular disorders, including muscular dystrophies and age-related muscle disorders, might be treated using gene therapy approaches that prevent or lessen the inhibition of muscle growth by myostatin. This could be achieved by the knockout of myostatin gene expression or by the overexpression of insulin like growth factor 1, which can increase muscle size and strength. To this end, transgenic mice were created from both dystrophic and normal mice by a single postnatal intramuscular injection of adeno-associated virus that resulted in the overexpression of a gene encoding the myostatin inhibitor protein follistatin. This single treatment enhanced muscle mass and strength in normal and dystrophic mice for more than 2 years. This therapeutic strategy warrants serious consideration for clinical trials in the treatment of human muscle diseases. One potential concern other than the safety of the viral vector is that gene therapy approaches that are intended to treat muscle disease by increasing muscle mass and strength might also be used for gene doping of healthy individuals to enhance athletic performance. Targeting systems, lipids. While the effectiveness of silencing RNAs for specifically inhibiting gene expression in cultured cells has been demonstrated on numerous occasions, it is difficult to efficiently deliver these RNAs to tissues in vivo. One approach to overcome this difficulty has been to chemically couple on silencing RNA at the terminal hydroxyl group with a sense strand RNA to cholesterol. The siRNA in question is complementary to an mRNA that encodes apolipoprotein B, a molecule involved in the metabolism of cholesterol. When the siRNA cholesterol complex is intravenously injected into mice, it is taken up by the liver, jejunum, part of the small intestine, heart, kidney, lungs, and fat tissue cells. Once this complex is inside the tissue, the sense strand is destroyed and the antisense strand binds to the target mRNA. With this approach, the level of apolipoprotein B was reduced by more than 50% in the liver and by 70% in the jejunum. This resulted in a significant decrease in the plasma apolipoprotein B level, as well as the total amount of cholesterol. This strategy is an important first step in the development of a method of therapeutically lower cholesterol levels in humans. A number of other molecules, including some long-chain fatty acids and bile acids, may be used in place of cholesterol to mediate the uptake of siRNAs into cells. A critical factor in mediating the interaction between fatty acid conjugated siRNAs and lipoprotein particles is the length of the fatty acid alkyl chain. Thus, docosahexaenol C22 and stearoyl C18 conjugates bind more tightly to high-density lipoprotein and subsequently silence gene expression more effectively in vivo than Laurel C12 and myristoyl C14 conjugates. Studies are underway to improve the delivery of lipid-conjugated siRNAs to treat a wide range of diseases. Bacteria that are normally found in association with various mammalian tissues and cells may be genetically engineered to produce therapeutic siRNA, short happen RNAs. The engineered bacteria may then be used as vectors to deliver therapeutic agent directly to the affected tissues. For example, a non-pathogenic strain of E. coli was transformed with the plasmid vector TRP containing the gene for the protein invasion which permits E. coli to enter beta-1 integrin positive mammalian cells, and the gene HLY-A, which encodes listerylation O, a protein that enables genetic material to escape from entry vesicles. In addition, the TRP vector carries an shRNA molecule under the control of a bacterial promoter directed against the mRNA produced by the cancer gene CTNNB1. As long as bacterium is able to enter target mammalian cells and release shRNAs, the shRNA may be directed against any specific mRNAs. E. coli cells act as a vector to transfer the shRNAs to where they are required, example cancer cells. This approach has been shown to work both for cancer cells in culture and with mice. With whole animals and bacteria can be administered orally. Now on to collagen. The protein polymer collagen, isolated from calf dermis, can be digested under acidic conditions by the proteolytic enzyme pepsin to form subunits of approximately 300 kilodalton each. These rod-like proteins, approximately 300 nm in length by 1.5 nanometer in diameter, are positively charged and therefore readily interact with and bind to the negatively charged siRNAs. These acylocollagen particles protect siRNAs from nucleus digestion and also can be injected locally for tissue targeting delivery of the siRNAs. For example, siRNA acylocollagen complexes have been efficiently delivered to tumor cells in mice and after injection can exist in an intact form for at least 3 days. Furthermore, in mice, siRNA acylocollagen complexes have been found to inhibit tumor growth in bone cells. This method of packaging siRNA promises to be both reliable and safe depending on the tissue involved. Antibodies. It has become relatively straightforward to generate monoclonal antibodies against nearly any targeted protein, to humanize those antibodies of these variable regions and then to produce them in heterologous host cells. Moreover, at the DNA level, it is easy to fuse the antibody gene with the gene for another protein. With this in mind, the gene encoding single chain fragments that bind specifically to a protein called ERBB2, which is found in the surfaces of breast cancer cells, was fused to the gene for the positively charged nucleic acid binding protein, protamine. The fusion protein binds to the surfaces of cells expressing ERB2 and its C-terminus carries the 51 amino acid long protamine, which readily binds to added siRNAs. In one of these in one test of this system, an anti-human immunodeficiency virus HIV envelope, Fab, and an siRNA that is designed to cleave the HIV gag mRNA were employed. Using cells in culture, it was possible to reduce the amount of secreted gag protein, the protein of the nucleocapsid shell around the RNA of a retrovirus, by greater than 70%. This system also works in vivo when the construct is injected earlier, intravenously, or directly into tumors. The hope is that by using a combination of specific antibodies or antibody fragments that direct siRNAs only to certain cells and siRNAs that selectively cleave specific target mRNAs, the system can be used to treat a wide range of diseases. Aptamers, the binding specificity to a target antigen that is a central feature of the functioning of antibodies, is also a property of aptamers. Thus, conjugating aptamers which bind to specific cell surface proteins to siRNAs that are designed to reduce the expression of certain mRNAs should provoke another method of targeting siRNAs to specific tissues or cells. Also, since both aptamers and siRNAs are chemically synthesized as RNA oligonucleotides, it should be simple and straightforward to synthesize chimeric RNA molecules that include both the binding specificity of an aptamer and an siRNA that targets a specific mRNA. For example, an aptamer that binds selectively to a prostate-specific membrane antigen found in prostate cancer cells was first selected. Then a 21 base pair siRNA directed against mRNA encoded by either of two genes that are necessary for prostate cells to survive was added to the aptamer sequence. Both activities, that is aptamer binding and RNAi, were maintained in the chimeric molecule. The targeting aptamer did not impair the ability of the siRNA to silence the target gene, and the presence of the siRNA did not affect the ability of the aptamer to bind to its target. The simple but highly effective approach should be amenable to treating a wide range of human diseases, provided that 1. Silencing specific genes in a population produces therapeutic benefits, and 2. There are surface receptors, usually proteins, that distinguish the target cell population and allow the siRNA to be internalized. Excuse me. And now on to the summary. A number of human disorders that result from the overproduction of a normal protein may be treated by using 1. Nucleotide sequences that bind to a specific mRNA and prevent its translation, that is an antisense oligonucleotide. 2. RNA sequences that bind and cleave specific RNA molecules, that is ribozymes. 3. Small RNA molecules that aptamers that assume a highly organized secondary and tertiary structure and bind tightly to a wide range of molecules including proteins, amino acids, and drugs. Or 4. A small double-stranded RNA molecules that direct the sequence-specific degradation of mRNA, that is interfering RNAs. These techniques may also be used to lessen or prevent diseases caused by pathogenic viruses and other disease-causing organisms. The greatest impediment to the development of nucleic acid-based therapeutic agents is the difficulty in delivering these agents to the target tissues. Initially, workers used virus-based delivery systems with some success, although some safety concerns exist in regard to the use of these vectors. Other approaches for the delivery of nucleic acid-based therapeutic agents include intravenous injection, local injection at the site of the pathology, packaging the nucleic acid into cationic liposomes, physical methods like electroporation, sonoporation, or hydrodynamic pressure, and conjugating the nucleic acid to another molecule, such as a lipid molecule, cholesterol, collagen, collagen, an antibody fragment, or an aptamer. The development of effective treatments for genetic diseases has been elusive because in many instances the appropriate gene product cannot be provided to a patient. However, when a normal version of a gene has been identified and cloned, it may be possible that either it or a cDNA derivative can be used to correct the defect in affected individuals. Viral and non-viral systems have been developed for the delivery of therapeutic genes. Viral vectors take advantage of the ability of a virus to penetrate a cell, specific cell, to protect the DNA from degradation directly to the cell nucleus. A number of viruses have been engineered for gene therapy applications. Packaging cell lines for some viral systems ensure that virtually no infectious viruses are present in a sample of vector viruses. Non-viral gene delivery systems include injection of pure DNA, bombardment of a target tissue with the DNA-coated particles, and cellular uptake of DNA that is enclosed within a lipid envelope. In addition, HSCs may find use as vectors for the long-term maintenance and gene expression of therapeutic genes in human cells. Generally, the major drawbacks of the current generation of gene therapy vector systems are immunogenicity, lack of cell specificity, inefficient gene transfer, and limited therapeutic gene expression. That's it about the summary on this chapter. Let's see if we can just cover up some questions too, or review questions. Now, for the review questions, how can antisense oligonucleotides be used to treat psoriasis? What are ribosomes and how can they be used as therapeutic agents? What are interfering RNAs and how might they be used as human therapeutic agents? What is an aptamer and how is it used as a therapeutic agent? How can antibody genes be used to confer passive immunity? How can interfering RNAs be delivered to specific cells? In developing a new nucleic acid-based therapeutic agent, how would you decide between antisense oligonucleotides, ribozymes, aptamers, and interfering RNA? And what are the key attributes of a therapeutic gene delivery system for humans? How can interferon response be, which is usually induced by a double-stranded RNA, be avoided when utilizing siRNAs as a therapeutic agent? What are some of the advantages and disadvantages of deoxyribozymes compared to ribozymes? How can the progression of age-related macular degeneration be limited using RNA therapeutics? Right, that's it for the questionnaire session as well. The review questions on this particular subject of nucleic acids as therapeutic agents. I'll be continuing on the next chapter of vaccines shortly. We'll give you the details on that. Till then, please prepare notes, take screenshots, and do as much as research as you can, and do the best you can to prepare well for the exams, of the NEET exams, of the Institute of Medical Sciences, the Council of the Scientific Industrial Research National Eligibility Tests, and other tests in India and overseas. Wish you all the best, but kindly do not commercialize any aspect of my lectures or the notes being shown to you, or do not take screenshots of the diagrams for the sake of sale or any commercial purposes. Please use them to the best of your ability to study and pass your exams. Wish you all the best. Thank you. Best wishes. Have a great day. Good day to all of you. Thank you. This is Dr. Bobby signing off. Bye.